This is the third part of the immune system. We are talking about adaptive immunity and cell-mediated immunity. So what we need to know is that once we start talking about adaptive immunity as opposed to innate immunity, we're talking about specific immunity, immunity that is responding to one pathogen only. Once we've been exposed to that pathogen, we know that our cells are able to basically identify that specific pathogen. It is much slower. It requires the support of the innate immunity to function, and it's got two parts. When we start talking about adaptive immunity, there are certain cells that we need to talk about, which hopefully we've heard before. B cells are synthesized in the mature in red bone marrow, and T cells are synthesized in the bone marrow but mature in the thymus, hence the term T cells. But they are very similar in what they do, just their locations are slightly different. This is why immature B and T cells are virtually indistinguishable. We have lymphatic tissue, which is consisting of our lymph nodes and all of the vessels that lymph flows through. T cells can migrate to and from the thymus using the lymphatic tissue. B cells remain in our bone marrow for a long time. T cells have T cell receptor sites that can attach to an antigen that is presented by another cell, such as a macrophage or a dendritic cell. This is how we start the process of reading what a pathogen is. B cell receptor sites can attach to antigens. They are made of 4 polypeptide chains held together by covalent bonds called disulfide bridges. Antibodies have the same construction as this. Where lymphocytes are activated, so when we see a pathogen showing up, essentially the cells that are in our lymph nodes are going to be activated first. We have lymph nodes in our tonsils, we have them in our face, we have them in our neck, we have them in our armpits, spleen, thymus. All of these are lymph tissues. So these are the 4 characteristics of specific or adaptive immunity. The cells are able to recognize self versus non-self. They can recognize your own cells versus a pathogen. The second one is specificity. Lymphocytes are tailored to combat a specific antigen, and so you have a huge diversity of B and T cells. That leads to the diversity portion. There are potentially billions of different antigen receptors on B and T cells that recognize billions of different antigens. And the last part is memory, their capacity to remember a pathogen once you are exposed. Like we talked about, once you're exposed once to chickenpox, it is very unlikely you will get it again unless you get shingles. How do we determine between self and non-self? And the way we do it is the major histocompatibility complex proteins, or the MHCs. These are cell surface proteins that are unique to specific individuals, namely you or someone else, unless you are a twin. There are 3 classes of proteins. The first class are on all cells. The second class are on antigen-presenting cells, such as B cells, macrophages, and dendritic cells. Basically, these allow us to identify our self cells. What is a major histocompatibility complex made up of? Basically 4 polypeptide chains, or we know conglomerates of protein that will come together to make a full protein. And they attach to antigens, which are small polypeptide chains. And so what we see here is that we've got our class 1, which are on all cells, our class 2, which are only on my immune system cells. And we can see in the third image, the antigen is going to bind to the MHC to dock, basically. They have this ability to attach to foreign materials and transport the foreign materials somewhere else. And we call these antigen-presenting cells, or APCs, and they're usually the phagocytic cells. As APCs are in the process of cleaning up an infection, bits of this pathogen are going to be displayed on the MHC proteins, which is how we end up with my self cell carrying around the identifier of the other cell, or the pathogen cell. T cells migrate to and mature in the thymus, and they interact with dendritic cells. And the dendritic cells are the cells that have the MHCs on them that will present the antigen protein. There are three possible outcomes for T cells interacting with dendritic cells. The T cell receptor binds to the dendritic cell with its own proteins, and apoptosis is induced and the T cell dies. The TCR weakly binds to a dendritic cell with the MHC with no proteins. The T cell survives, and the TCR does not bind to any MHC protein. Apoptosis is induced and the T cell dies. What that means is that the only way that T cells are going to survive is if it weakly binds to a dendritic cell with MHC with no proteins. So what that means is that the surviving T cells can recognize MHC proteins that have some foreign protein in their cleft. T cells ignore MHC proteins without any foreign material in the cleft, or healthy cells. How do B cells recognize this? So B cells mature in the bone marrow. If the receptor site on the B cell matches a surface protein found in the bone marrow cells, apoptosis occurs. The B cells that survive are those that are going to ignore the body's own surface proteins, but are recognizing foreign material. Not all B and T cells are destroyed. The ones that do survive are suppressed, so they don't react with their own body tissues. We'll talk about this a little bit later. But then we end up with like our immune system attacking our own immune system, which is a problem. When B and T cells mature, they wait in a secondary tissue basically until they are necessary when we see a pathogen getting involved. So that's the first part of what makes this immunity so special. The second and third, the specificity and the diversity of the immune system, refer to how it can respond to a very specific antigen. This lies in the receptor sites on the B and T cells and the production of free-floating antibodies. B cells produce proteins called antibodies that interact with pathogens. The basic structure is very similar to the B cell receptors. The B cell receptors on the left, that would be what docks with the dendritic cell. And on the right is the antibody that is produced, looks very similar. This talks a little bit about the genes of the receptor sites and the antibodies. We're not going to go into that today because we're not talking about genetics. We'll move forward. The second part of the adaptive immune system or specific immunity is cell-mediated immunity. The first part, it's divided into two parts. Cell-mediated immunity selects T cells that recognize and destroy infected body cells and cancer cells, which is what we've been talking about. Humoral immunity selected B cells produce a copious amount of antibodies to fight pathogens, and so this we see is where the separation of T and B cells shows up. First, T cells show up and try to destroy any infected body cells and cancer cells, and then the B cells produce antibodies to fight the pathogens. The cell-mediated immunity needs the support of cells and the cells involved in the innate immunity. So dendritic cells, if we remember, are part of innate immunity, and so are macrophages, and they're used to engulf the antigen and present the antigen to the T and B cells. And this, in turn, allows the T and B cells to recognize what the antigen is and start to become very specific to the antigen. This is how it happens. The macrophage and the dendritic cells arrive at the site of infection. This is innate still. This is very general, and they go through phagocytosis. They eat the pathogen. These cells, macrophages and dendritic cells, have MHC1 and 2 proteins, right? These receptors on the surface. The antigen combines with either the MHC1 or 2, and the complex migrates to the surface so that these cells are antigen-presenting cells. The antigen-presenting cell migrates to secondary lymphoid tissues. This is where the B and C cells are going to hang out. The first thing we go through is cell-mediated immunity. And so the dendritic cell or the macrophage, both of which are APCs, are going to search for a match with a T cell. If the matches with the MHC1 protein, the T cells become a cytotoxic cell, also known as a memory cell. If the MHC2 protein makes a match, the T cell is destined to become a helper T cell, and memory helper T cells, which we'll talk about the memory T cells a little bit later. As a little bit of a reminder and a background, dendritic cells are found in the skin and other tissues that interact with the environment, which is why they're so good at becoming APCs and are involved in bringing the antigen to the T cells. Take a minute to pause and mull this over. See if you can answer this question. While the appropriate T cells are being selected in clones, body cells are becoming infected. Bits and pieces of the pathogen end up on the MHC1 proteins of all different types of body cells. The body cell is tagged as infected, and selected cytotoxic cells recognize that and attach to the cells. And remember, now my T cells have become cytotoxic cells. Once it's attached, my cytotoxic T cells secrete something called perforin, and that's important. Perforin makes pores in the membrane of the infected cell, and the cytotoxic cell also secretes granzymes, which causes the infected cell to die. So there are two different enzymes which are secreted and cause the infected cell to die. These two things are secreted by the cytotoxic T cells. This is another image of how that happens. Feel free to review it if you'd like. Regulatory T cells help halt the immune response. We don't really know how they're activated actually, but what we do know is that they are going to stop any more B or T cells from being activated, and it slowly stops our immune response, and the activated immune cells are going to die off eventually. They have a very short lifespan, so after they're no longer needed, they're going to die off pretty quickly.